So hello and welcome to Bioaffairs. Today I am here again to make your classes very simple. So today I'll be talking about bacterial shape. You know there are different kinds of shape regarding the bacteria. Some are rods, some are Coccis, then comma shaped, then spirillum, filamentous. There are several types of shapes depending on different bacteria. Have you ever wondered why this kind of shape forms? What is the mechanism? Why the Cocci is Cocci, not a rod? Why the rod is rod, not a Cocci? So today we'll be disclosing this mystery, or we'll be knowing the mechanism why it is Cocci, why it is rod shaped, and also we'll talk about comma shaped bacteriums and some other types of bacteriums, why they are spirillum or fibrillum, any kind of. So let's talk about this. So do you know the most common shape which is found in nature of bacteria? That is the rod shape. This is the most common shape of bacteria. Now what is the most simplest shape of bacteria? Can you tell me? You have 5 seconds. Okay, it is the Cocci shape. The most simplest shape is the Cocci shape and most common is the rod shape. Now start this with the Cocci. So the spherical shape or the Cocci shape is the same thing. Now remember this. The Coccis, as you can see in this picture over here, the Coccis are always the septum formation. In my previous class, I have told you about septum formation, cytokinesis, binary fission processes. So please go to that class. You will be finding in the I button, and then come to this class. It will be easy for you to understand. So here the picture depicts a Cocci shape. Now you can see the shady area in the middle. Okay, in that middle is showing you the septum formation. Okay, that septum formation is always will be there in the middle. That you should remind, that you should remember. In case of spherical bacteria, septum formation will be in the middle, and from there it will enlarge, it will elongate, and then it will divide to daughter cells. So progeny cells will contain one old hemisphere and one new hemisphere. What is hemisphere? Hemisphere means when the two cells or two Coccis are dividing, as you can see in the picture, that time what happens? In the middle, new peptidoglycan is preparing, new peptidoglycan is synthesized. So in the other sphere, if you divide this sphere, suppose this is the parent cell, consider this parent cell, it is a single sphere, then the two hemisphere, if you divide in two hemisphere, left and right, so the left is the old peptidoglycan, and the right one from the middle, new peptidoglycan synthesized. So this is new hemisphere. Okay, the one old hemisphere in the daughter cells, as you can see here, and one new hemisphere. I think I am clear. Okay, now this old and new hemispheres will be always, what I say, constant during the Cocci division. Okay, that is why a Cocci or a sphere shaped bacteria, when it's divided, always will be in Cocci shape with one old and one new hemisphere. You understand the hemisphere? That is the two. You cannot say pole here because it is a circular thing. So I have told you hemisphere. Okay. Now coming to the rod shaped bacteria. Why it is always rod shape during division or formation of daughter cells? So here you can see three stages actually, three steps or three stages actually. So first there will be sidewall elongation before the division. So this, remember this, during growth and comma prior to division. So division has not yet started, but growth is taking place, means it is increasing its cell components, cytoplasms, ribosomes, cellular size, and that I am telling you, sidewall elongation. Sidewall elongation is very important thing here, which is assisted by MreB homologs. This is a protein, MreB proteins, which are analogous to eukaryotic cytoskeleton proteins. Okay. Now this is helping in sidewall elongation. So if you consider this a bacteria, as you can see in this picture, the sidewall basis is increasing. If you can imagine the 3D structure of a bacteria, this is not hollow here, actually it is sidewall. So sidewall elongation of these MreB filaments will be just putting there in the sidewall basis, and there will be elongation or synthesis of their sidewalls, all total sidewalls. So what will happen? The cell will enlarge in its, cell will swell or enlarge its shape. Suppose the parent bacteria was 1 micrometer. Then suppose during this step, sidewall elongation, this elongation process, the bacteria will be around 1.2 micrometer. I am just for imagination I am telling you 1.2 or 1.5, whatever you want to understand, as you can see in this picture. Now during the second stage, coming to the second stage, where pre-septal elongation, PSE. Now PSE is not a word, you will not find anywhere in a book. PSE I have made it now. Just during PSE, pre-septal elongation means septum is not yet formed. Till now no septum formed, during which FtsZ, it is a tubulin homologue of cytoskeleton protein that is present in the eukaryotic cell, but in bacteria it is FtsZ, which what in the previous class I have already told about this FtsZ. So please go to the previous class, then come here. Now FtsZ polymerization will cause Z-ring formation. Okay. Now this Z-ring formation, so new cell wall, this Z-ring will form in the middle of the elongated cell. Now you have not the parent cell, but it is more enlarged cell. So the cell, if you consider this a cell, suppose this is a cell, suppose this pen is a bacteria, okay, it is a parent bacteria. Now during enlargement it became like this. Now the parent bacteria become this. Before division, before septum formation, I am telling you. So before this septum formation, the parent bacterium, after the enlargement or elongation, before pre-septum formation or septum formation, it became like this, not yet divided, remember that. Okay, and this at this stage, this FtsZ polymerization causes the Z-ring formation. So Z-ring will form right at this place in the middle. Okay, so where it is forming? It is forming in the enlarged bacteria, in the middle of the enlarged bacteria, where FtsZ units will just add to form a Z-ring. Z-ring is for what? For what? It is for septum formation. Now it is forming in the middle. Now this enlarged cell from the pre-septal elongation will now go for division or cytokinesis or cell wall formation. What will happen along the Z-ring? So when it accumulates, the Z-ring formed, septum formation will start and it will divide the cell from the middle. So as you can say that the cell is divided from the middle, but actually it is not the original shape of the cell because it was much more less elongated. Now it is elongated from 1 micrometer to it is 2 micrometer. If the parent cell is 1 micrometer, it became 2 micrometer during division, and from there again in the middle there is a Z-ring formation, septum formation, became 1 micrometer, 2 daughter cells or progeny cells. So now progeny cells, as you can see, will have one new pole again and one old pole, like the one new hemisphere and one old hemisphere regarding the Cocci, and here we will say it as a pole because I can see in the picture that the middle of the cell is now become the end of the progeny bacterium or the daughter cell of the bacterium. So as you can see from the middle here, so hemisphere is forming here. Okay, so it became a pole. It became a pole for one bacterium and a pole from another bacterium. That is from daughter cell pole 1 you can say for another daughter cell or daughter cell 2, this is pole 2. So that is the thing you need to understand. Why the Cocci is Cocci and the rod is rod always. So these things, these phenomena happen, and these proteins are very, very important. MreB, this cytoskeleton homologue, is very essential in forming the sidewall elongation during the division of their cellular components. Now I told you I will talk about comma-shaped bacterium. Now you can see here it is a comma-shaped vibrioid called caulobacter crescentus. Okay. Now you can see in the picture 3 proteins. Already MreB I have told you, FtsZ I have told you. Now here you will see a new protein, that is crescentin. Okay. So crescentin is the new protein you are listening. It named after the bacterium. So why it is comma-shaped? Actually, this crescentin localizes along the side wall, as you can see in the picture. This MreB and FtsZ is in the middle at the upper hand, and the side wall contains this crescentin in the lower portion, you can see. Now what is the function of this crescentin? MreB and FtsZ is normal functioning. They are helping in peptidoglycan synthesis. Already there are synthesis of peptidoglycan. But in case of crescentin, its function is to slow the process of peptidoglycan synthesis. Suppose in 1 minute, the natural addition of peptidoglycan is 10 nag or nam units. Suppose this is not the case. I am telling you to make you understand. So 1 minute, 10 nam units per 1 minute. It is the normal rate of peptidoglycan synthesis. But due to the presence of this crescentin in the sidewall, 1 nam unit is being added at 1 minute. So whether or when it was required to add 10, now it is adding 1 only. So what will happen? The cell wall composition will not be enough or will be less. So it will form a curve because the filling is not there. The area which should be filled with 10 nam units per minute is now filled with only 1 nam unit per minute. So that is why the area is not filled properly and it became a curved shape like this. You can see it is a vibrioid with comma shape. So this comma shaped structure comes due to the crescentin protein in case of caulobacter crescentus. Okay. Now some bacteria may have, mostly Vibriod have this kind of proteins which slowing down the peptidoglycan synthesis process in a particular wall, along the wall. That is why a comma shaped structure is formed. I hope you have understood this class. Now I will talk about some important other shapes which have different mechanisms other than this form, because all bacteria are not Cocci, all bacteria are not rod, all bacteria are not comma shaped. So there are other shapes also. So I'll give you some idea about this other shapes. Okay. Like the spirochete Borrelia burgdorferi, which causes Lyme disease, relies on the periplasmic flagella to confer its spiral shape. So here the periplasmic flagella is conferring its spiral shape, because we know spirochete is a spiral shape. But why it is forming? Because of the presence of periplasmic flagella, which gives it a unique shape. Okay, taking another example, spiroplasma, a citrus pathogen. Mostly it is affecting citrus plants. So these bacteria do not contain any kind of cell wall or flagella. So why this kind of shape it can show? How it can show? So it generally relies on contractile cytoplasmic fibrils to give the bacteria a spiral shape. So in case of spirochete Borrelia burgdorferi, the spiral shape is given by a periplasmic flagella, whereas in case of spiroplasma, this spiral shape is given by cytoplasmic fibrils. So these two mechanisms are different and also different from other mechanisms. So here I have given you some examples of most important things, bacteria shape, which will give you some idea how these things work and the mechanisms. And if you really like my classes, give a big thumbs up and don't forget to subscribe my channel Bioaffairs. This type of subscription and likes give me the enthusiasm to prepare more futuristic classes for you guys.